Exciting to be here at this conference and the stimulating program here at the KATP. In my talk today, I will focus on theory and discuss how condensates can organize chemical processes in original ways that can be used by cells. And this work is stimulated by a very collaborative environment in Dresden, where we have for many years collaborations, particularly with Tony Hyman, and on the theory side we have collaborated for many years with Christoph Weber. And for the work I'm going to present today, I'd like to highlight collaborations with Tyler Harmon, with Jan Bruges, with Simon Alberti and Nagaria Chapiti. And I'd like to highlight two very strong postdocs, Ryota Takaki and Rakesh Das. So cells exhibit many different types of condensates that provide distinct biochemical environments that organize biochemistry in space. And our thinking about these structures and their properties is guided by the physics of phase transitions and phase separation. And this happens in a very non-equilibrium environment. And this has motivated us for a number of years now to study the interplay of phase separation, of condensation with chemical processes, in particular non-equilibrium chemical processes, so to understand the emergent behaviors that come out from this interplay. And as it is non-equilibrium, it can lead to interesting spatial-temporal organization. So some basic ideas are that droplets can act as chemical microreactors. They can locate and regulate chemical processes. But of course, the phase separation and the droplets themselves can be chemically regulated and organized. If we first look at a passive system, that's a system that would relax to a thermodynamic equilibrium. Phase equilibrium and chemical equilibrium must be consistent with each other. And this can be illustrated in the following way. So we can describe phase equilibrium by binodal in the phase diagram, where tie lines connect coexisting phases with their composition. Chemical equilibrium can be described by a manifold in the same space. In this case, for example, this red line corresponding to chemical equilibrium points, and the consistency of both requires that the chemical equilibrium line intersects the bimodal in points that are connected by tie lines. And this thermodynamic consistency leads to a relationship between equilibrium constants of chemical reactions, which I call K here, with reaction index alpha. They are different in the coexisting phases 1 and 2, and their ratio is related to partition coefficients for species K, where in the exponents we have stoichiometric coefficients. Now, in active systems, which are maintained out of equilibrium, which are coupled to chemostats, this relation can be broken. In this case, we can have situations where phase equilibrium and chemical equilibrium are no longer consistent. So those chemical reactions will not equilibrate at phase coexistence. And these are active systems that cannot relax to equilibrium. And these are the sort of the theme of systems we are interested in many different contexts and ways. And in my talk today, I just want to focus on two recent themes of our interest. In the first part of my talk, I will discuss how one can use molecular switches and molecular modifications to create identity of droplets in a non-equilibrium setting. In the second part of my talk, I will discuss condensation of DNA together with proteins, and how such condensation can be modulated with active processes such as DNA loop extrusion, or in the context of DNA double-strand breaks. So let's come to the first part, which is work in collaboration with Tyler Harmon and Jens-Uwe Sommer in Dresden. And the basic question is how to think about many different types of condensates that coexist in the same volume of a cell. And one way to think about this problem is to notice that multi-component mixture can exhibit multi-phase coexistence according to Gibbs phase rule. So as an illustration here, in a ternary system, we can have a region where 3 phases coexist, and this allows in principle to have 2 different types of droplets existing in the system at the same time. However, in a cell, equilibrium is usually something one can maybe assume locally, but not globally, so such a picture may be limited. And I want to discuss an alternative picture where we have essentially rather unspecific interactions which generate phase separation, but molecules that participate in phase separation can be modulated or modified. So I have these green and purple types of molecules called A and B. Both of them can phase separate from the background, but they can be modified by an enzyme. I use here the simple example of kinase and phosphatase as an example, but any type of modification would work for this scheme. So the kinase turns B to A, and the phosphatase turns A to B. These are non-equilibrium reactions. And the idea is that even though the phase separation is quite non-specific, we can use such non-equilibrium setting when the enzyme tends to partition in an environment which is rich in its own product. So the kinase would tend to partition into an A-rich phase, the phosphatase into a B-rich phase. And then we can use this sort of enzymatic specificity to generate identities of droplets. We call this droplet differentiation. And the simple way to illustrate it as an example is to use a Flory-Huggins model with 2-phase coexistence, where this chi parameter describes phase separation of all these components from the background solvent, and we have a small perturbation that leads to partitioning of the enzymes together with their reaction products. Now, this perturbation here is not enough to generate multi-phase coexistence. The system has essentially 2-phase coexistence at equilibrium. And if we now look at this dynamics, we write down the Grahn-Hilliard equations with reaction terms. We have sort of here Michaelis-Menten type of enzyme kinetics. And the system has a behavior that depends on the reaction kinetics, on the rates of these reactions. If this rate is small, or if the system is at equilibrium, then it will go to a state which is symmetric, where we have always A and B together in equal composition inside a droplet. And if we ramp up this rate K, bring the system out of equilibrium, these null lines change, and this initially stable fixed point becomes unstable. We now have differentiated droplets corresponding to these 2 different stable fixed points. So the system then, as a function of the reaction rates, undergoes instability and differentiates in 2 different droplet types, and the instability condition is written here. Just to show you what this looks like, if we start with a phase-separated droplet which contains both A and B, and we run this system, then it differentiates and splits in different droplet types. And this now sort of 3-phase coexistence is purely non-equilibrium. It's generated by these reactions. If we just quench the system with the phase separation, it first does the usual phase separation kinetics, and then this differentiation takes over. And what you can see in this example is that the 2 types of droplets ripen independently. So the purple and the green show separate Ostwald ripening in this case, and that's nicely differentiated. So this example shows that one can use sort of rather non-specific interactions for phase separation, and then use specific enzymes to create, or specific enzymatic processes to create out of equilibrium a multi-phase coexistence, realizing condensates of different type, and we could use more than 2 in such a setting. So I then come to the next part of my talk, which is completely different type of question, but it's also about condensation and active processes. But in this case, condensation of a polymer that condenses together with proteins. And that's situation that is relevant to the spatial organization of DNA or maybe chromatin in a cell. And I will be discussing loop extrusion and double-strand breaks. Now let's first come to loop extrusion. This is work done with John Bruegges and with Ryota Takaki. So loop extrusion is a process where a motor interacts with a DNA molecule and pulls on the DNA to form a loop, which is illustrated here. These are called structural maintenance of chromatin complexes, and they have been suggested or shown to be related to the spatial organization of chromatin, which can be revealed in DNA contact maps, where the probability of contacts at different positions along the DNA is shown with this color code, and these squares somehow correspond to segments that can be reasonably large, while many contacts occur within but not outside, and such a loop can naturally allow such contacts. Now, another way to organize such a polymer in space is to use condensation. Of course, one motivation is to think in terms of transcriptional condensates, but one can, in many cases, also have situations where just proteins condense together with DNA as a polymer to form protein-DNA condensates. This is something which we've been studying also with John Bruegges in recent years. He has a very beautiful experimental assay where DNA is attached to a glass surface at 2 points, so it's somehow maintained at a certain end-to-end distance, and this allows to observe in a microscope the state of the DNA. And if a protein is added, these foci form. In this case, this is a transcription factor called FOXA1, and one can see that both the DNA and the protein signal are show a foci, so there's a co-condensation of the two. So this is a schematic drawing of this, and this happens while the DNA is stretched. One can describe the physics of this by having a droplet model for these condensates with a bulk term, a surface tension term. LD is the length of DNA inside the condensate, which is then in the condensate proportional to the volume. And when we have to add to this the polymer stretching free energy, have the balance between the stretched polymer pulling on the condensate. So one can use this assay to measure now these parameters for these different proteins. And just to give you an impression of how the system behaves, if one changes the end-to-end length, it's short, then we get a large condensate. If we have a longer DNA length, then we pull on the condensate and make it shorter. And beyond a certain point, there is no condensate anymore. There's a first-order transition between having no condensate and condensation. And this can be compared to experiments. Now we'd like to understand what happens in such a setting when we have loop extrusion. And to do that, we use Brownian dynamic simulations. It's done by Ryota Takaki. So it's a simple coarse grain model for a polymer where we have DNA, we have protein particles that can condense, and we add a motor that can extrude a loop by stepping from monomer from bead to bead and pulling a loop in this situation. And we can now in this model study both loop extrusion and condensation and their interplay. So this is an example of loop extrusion studied in such a simulation. Now here we can see one of these motors has pulls slowly a loop out here. In this case, there's no condensation. These white dots are beads that could have properties to condense, but here they are not condensing. And we are creating this loop, which reveals that while the DNA is under tension because it's held apart at the ends, the loop is tensionless, and it provides a region of high density, and with many contacts also. But we can now change the properties of our beads that float around and have them condense with the polymer. So we look now at loop extrusion happening in a situation where proteins can condense. And now you can imagine that these regions where loops start are ideal nucleation sites for a condensate. And you suddenly see them form little spheres. And now the loop extrusion continues and feeds the condensate, which grows. And also the different condensates that were nucleated are pulled together by the loop extrusion. So in the end, this process reliably produces one beautiful condensate, which is exactly at the position where the loop was meant to be extruded, which is a sequence-dependent process. So these machineries, they find certain region on the sequence to extrude the loop. In this case, they nucleate a condensate of the right size at the right place. So this shows that loop extrusion is an ideal system to nucleate condensates because it's a tension-free region of high density of chromatin as compared to the rest. So it's thermodynamically favored to nucleate there. The loop extrusion itself accelerates the condensate dynamics because it now grows fed by the motor. The loop extrusion also stabilizes condensates because they're now more stable with respect to forces pulling on the polymer. And the loop extrusion determines condensate position and size. And it creates sort of contact maps that are really quite nice and clean. So in the last 5 minutes, let me come to the last part. That is another problem of DNA organization by condensates, and that's a collaboration with Simon Alberti. And the problem here is, how does a cell respond to a catastrophic event where DNA breaks? In fact, a double-strand break is essentially very dangerous, potentially lethal. It needs to be repaired, and in order to do so, the broken ends must not be allowed to move away from each other. They have to be kept close together. This happens with an assembly of proteins at the break, form a condensate. After that, this allows to recruit all the machinery for repair. But the question I want to discuss here is how to keep the ends together. And this happens with a fast assembly of a condensate of a protein called PARP-P1. This can be studied and has been studied by Simon and co-workers in the same assay that I described before, in vitro, where one can use a restriction enzymes to break the DNA, and in the presence of the PARP-P1 protein, a condensate forms that holds the ends together, even in this in vitro setting. An important information about what's going on here comes from experiments that show co-condensation of PARP-P1 with short DNA stretches, and it reveals that it requires DNA ends for this condensation to happen. So if DNA ends are blocked, this system is essentially not condensing. And we therefore use a simple model, again in our Brownian dynamics picture, where the protein that condenses has two possible states: a soluble state A, and a state where its phase separates with the DNA B. And the transition between the two is catalyzed by the broken DNA end. So the DNA exists also as normal segments as an end, and the soluble form, if it meets the DNA end, is quickly transformed to the form B, but it only happens if there's an end. So the specificity comes from the catalytic properties of the DNA end. If we just have a stretched DNA molecule and it breaks, the two ends will move apart. And now the game here is to nucleate and grow a droplet faster than these ends can move apart, and to fuse the two ends together. And this can be studied in these Brownian dynamic simulations. Here you see the distance increasing with a break, and this would be a case where we have a fast enough catalysis. So let me show you examples. There are 4 cases. The upper 2 are fast catalysis. The lower 2 are slow catalysis. The left side is weak segregation in terms of phase separation. The right side is strong segregation. And of course, the lower right is the inefficient case where the DNA breaks and the ends are lost. Here we have condensates at the 2 ends forming, but they were forming too slowly. And the strong case is here when we have strong enough catalysis and the condensate holds the ends together, and we can see the diagram where this will happen. Maybe just as last comment, this requires the end to move slow enough that the droplet can grow and at both ends droplets can fuse. Interestingly, if we study the kinetics of an end as it retracts after the DNA breaks, this is for a strong stretching, weak stretching. The solid line is with enzymatic activity, the dashed line is without. We see that the enzymatic activity at the end actually slows down the retraction and helping the ends to stay together. This is shown in this example. And this has to do with the condensate exhibiting friction, preventing the end from moving fast. So with that, let me just briefly summarize. I've shown you that phase-separated droplets can spatially organize enzymatic processes and active processes in interesting ways. I've shown you as an example that condensate identity can be self-organized by active processes using molecular modifications. And I've shown you sort of interesting biochemistry enabled by condensate in the context of DNA end capture and DNA loop extrusion. And with that, I thank you for your attention.
Thank you. Questions?
So those timescales at the end of the story seem to be crucial. Do you have estimates for what those look like inside a cell? What's the diffusion time for these PARP1 proteins, and what's the retraction timescale for, chromosomal DNA?
So I don't know these timescales in the cellular context, and we're looking at the conditions in the in vitro system, which is sort of easier to manage, but it's also ongoing work. So we're still working out the timescales. It's something which we cannot really respond in detail yet.
Maybe as a follow-up, I mean, it's not only timescales, but also the concentration of the protein must matter a lot, so the condensate grows quickly enough.
Yes.
Have you done overexpression, underexpression studies like that?
So the protein is extremely abundant in the nucleus, and the surprising thing is that it is extremely abundant, and nothing happens unless the DNA breaks. So the system is like a loaded spring, ready, and it must not fire in the wrong situation. It must be extremely specific to the end break.
It keeps amazing me that these PARP1 condensates can keep the ends of the DNA together. I was wondering whether there's an estimate from the experiments or from the simulations, what kind of forces can you actually mediate via these condensates under realistic balance of interactions?
So I think in this essay we know what the forces are. I would have to look up the numbers because we know the polymer forces given the spacing.
Exactly.
I guess in the cell it's much harder to know what the forces are. Yeah. But it's quite surprising to see that it's not so difficult to, of course we have to push the rates high enough, but it's not implausible. Yeah. No, it's quite surprising how robust it is. Maybe Simon has some comments on that, I don't know.
Yeah, we measured it, but I mean, I don't recall the numbers right now.
Yeah, I also don't recall numbers. Yeah.
My question is along similar lines. Specifically in the simulation, do you include hydrodynamic interactions?
No.
And if you do, would that help you in this case? You could add that material along?
I have to think about that. I don't think it helps, but I have to think about it, yes. Mm-hmm.
Any more questions? Yes.
Thanks for the talk. In the first part for the droplet differentiation, have you thought about what if the kinases and enzymes were actively being produced and degraded themselves? Do you get oscillators or cycles between differentiation and de-differentiation, or is that not?
I think the whole setting has many possibilities, and I think Christoph has worked on oscillatory cycles in related systems, so it's an interesting question to look at oscillations. So we have been particularly interested in sort of differentiation in terms of how to generate specific identities given not very specific interactions for phase separation. And I think combining enzymes with phase separation gives you the biological specificity that you observe. It's sometimes hard to understand.
So the contact maps of the DNA looked very different in the loop extrusion versus the loop extrusion plus condensation model. So which one is the one that agrees with experiments? And what would be the meaning of having much more DNA-DNA contacts, like biologically?
Yeah, of course, this is not really a model for chromatin in a cell. It's just a model for the experiment in vitro. So I wouldn't want to really compare it to experimental data because there are histones and many other things around. But if you use loop extrusion and condensation, you get a much more compact structure, which generates a more, in the in vitro system or in the simulation, a more clear structure that looks like a TAD, topologically associated domain, while a fluctuating loop does less so. But that's maybe not the right comparison to a cell, so I would be cautious with that. But it's a way to discuss the in vitro experiments and to get some ideas about what might be important in a cell.
Hi. I have two questions on the loop extrusion portion. The first being, you expressed it as that loop extrusion was really helping nucleation. But if you had a similar tension-free polymer, would that's without you just the polymer is free, you would still expect this to condense, and should I view the loop? So like if the chromosome is instead just not under tension, would this also be expected to phase separate, and we could view loop extrusion as really just putting the chromosome under a state of zero tension for some fixed portion?
That DNA in the chromatin is under zero tension. So I think there could be many reasons why, if you look at a DNA strand and you cut it, that it would tend to move apart. So I think generically it should be under some tension, but I don't know what the tension is. And the assay, of course, has naturally tension in it, and it helps me somehow to discuss an extra quantity that it is the mechanical tension on DNA and its role. But I think zero tension is not the exact condition for chromatins. But I don't know what the condition is. So I think having the loop extrusion creating a region with low tension is relevant also to chromatin.
Okay. Yeah. And then I was wondering too if there's another nucleation barrier possible in this problem, which is if you have two sites that have condensed, bringing them together. And this seems like this could be a very large nucleation barrier as well. And loop extrusion could also solve this problem that you just zip them together.
Clearly, loop extrusion leads to coalescence of different condensates, also in the simulations, yes, in a very natural and robust way, yes. Mm-hmm.
I mean, in eukaryotes, chromatin is attached through a very big protein called the link complex to the cytoskeleton, to microtubules and muscle cells, actomyosin. So that could be one source of tension on the DNA. Yes. And people have actually seen that if they use blebbistatin, things change in the nucleus. And I sort of agree with what you said about tension.
Okay, it would be interesting to know what the values are.
Okay, one last question. Do you want to fight?
Actually, to follow up Ben Schuler's question, so we've actually directly measured the forces by single molecule pooling. So this can range from about 10 piconewton all the way to strand separation forces. Are these kind of ranges that are relevant for your simulations, the forces?
I guess below strand separation forces, but I have to check up the exact numbers, but I would say tens of piconewton in principle, yes. But
yeah, that's actually what we measure.
Yeah.
Yeah.
Mm-hmm. Yeah.
So maybe David can ask this question while Will is setting up.
I have another question for the third part. So you said that the PARP1 or whatever it's called is at very high concentration, but then that means that it could also nucleate spontaneously, or is that prohibited?
It does.
It could, you need to ensure that these condensates don't nucleate spontaneously,
it
right?
needs a conformational change to nucleate.
Okay.
And that's why I need this enzymatic property of the DNA end. And it's so specific that it only happens if there is a DNA end. And you see that in these co-condensation experiments of Simon and co-workers, where if you put short DNA with blocked ends, you don't condense. Well, if some ends are accessible, they do. And that's why we're having this catalytic step in the model.
Okay, let's thank our speaker again.